Good day and welcome to Herantis Pharma Research and Development Day. We have an exciting afternoon ahead of us with presentations from both internal and external speakers. They will talk about how the Herantis drugs Cdnf and Cdnf act on a key system in the body called Proteostasis. We'll see new and exciting human disease biomarker and biological data for Herantis drugs, and how those biomarker changes correlate with clinical improvements in motor function, brain, and brain imaging. We will hear about links to recognize Parkinson genetic mutations, and we'll also get an overview of the already established safety of Cdnf in humans. And of course, we will end today's session with a Q&A. But first, let's begin today with a video that perfectly illustrates what the company does.
Parkinson's disease affects 7 to 10 million people worldwide, and as the population ages, this is expected to increase. Parkinson's is a neurodegenerative disease that results in deterioration of motor and non-motor functions following death or dysfunction of the dopamine-producing nerve cells in the brain that are required for these functions. Cell death can occur when a key regulatory mechanism, proteostasis, becomes disrupted. Proteostasis is responsible for synthesis, folding, and degradation of proteins within cells, and is important for cell function and survival. Herantis Pharma has pioneered a therapy that aims to stop the progression of Parkinson's disease by restoring proteostasis using therapeutic human cerebral dopamine neurotrophic factor, Cdnf. Cdnf is naturally found in the endoplasmic reticulum (ER) of cells and protects cells from ER stress and the subsequent unfolded protein response, UPR, that disrupts proteostasis. Cdnf does this by interacting with proteins in the cell and adjusts UPR signaling. However, levels of neuroprotective molecules like Cdnf decline as we age, making the brain more vulnerable to degeneration. To combat this, therapeutic Cdnf can be administered to patients with Parkinson's disease or other neurodegenerative diseases to support the function of dopamine-producing nerve cells. Cdnf has the potential to stop the progression of these diseases and allow patients to maintain their quality of life.
And now it is time for me to hand over to the CEO of Herantis Pharma, Craig Cook.
Thank you, Kaiser. Good day, everyone, and welcome to the Herantis R&D Day. Thank you for joining. My name is Craig Cook, and I'm the CEO of Herantis, and we are delighted to be here and able to give you an overview of the company and our programs. We thought it was important to hold this R&D Day because over the past 6 to 12 months, we've generated some fairly compelling and quite significant data as it relates to our CNS assets. And we wanted to share that with our key stakeholders and other interested parties. We also wanted to do this in the context of Parkinson's disease generally. So we've invited some eminent external speakers to provide that context, and we very much appreciate their participation. The agenda for today, after my brief introduction, we will have Professor Blum giving us an overview of Parkinson's from a population perspective or a pandemic perspective. Dr. Henry Huhtinen, our CSO, will then provide an update and overview of our Cdnf program. He will be followed by Magnus Sjogren, Dr. Magnus Sjogren, our CMO, who will talk about our Cdnf program. Mr. Russell Patton from the European Parkinson's Disease Association will then give us the patient's perspective of Parkinson's disease. And Professor Hakan Vidner will provide the physician perspective and shape of things for the future as it relates to Parkinson's disease. I will then finish off, and after that we'll take Q&A. So on that note, and to set the scene, I'm going to give a brief introduction to the company before handing over to the next speakers. So Herantis is headquartered in Helsinki. We are listed on Nasdaq First North in Finland since 2014 and in Sweden since 2019. And over the past year, we've made some key data-driven decisions to focus on our CNS assets. And as a result, as of 2021, we are a pure-play CNS biotech company. Our research and scientific focus is on disease-modifying therapies. In other words, slowing, stopping, or even reversing the pathology of disease. Our disease focus is on Parkinson's disease and other neurodegenerative illnesses, where we're looking to bring treatments for those illnesses into the 21st century. And over the past decade or so, Herantis has made intensive and comprehensive R&D efforts and research to develop our CNS assets. And I think during the sessions that you see today, you will see the fruits of those efforts in terms of compelling data sets as it relates to clinical, biomarker, imaging, as well as genetic data, which are all key aspects of Parkinson's disease and their treatments with Cdnf and ex-Cdnf. Our pipeline comprises two assets: Cdnf, which stands for cerebral dopamine neurotrophic factor. That's a natural protein that occurs naturally in the body, and the natural role of Cdnf is to protect neurons in the body. And Herantis is looking to take that natural ability and harness it as a treatment for Parkinson's disease and other illnesses. We completed a phase 1 at the end of last year. The Cdnf in that study was administered via invasive surgical administration directly into the brain. So moving forward, we're moving away from the need for surgery and instead focusing on nose to brain or intranasal administration by a spray and subcutaneous or skin injection routes of administration. Our second program, ex-Cdnf, is a chemically re-engineered version of Cdnf, and we recently achieved an important milestone in that program where we selected the lead compound that we will be taking into further development. So that brief introduction hopefully provides a useful framework for the presentations that follow. And I'm now going to hand over the mic to the next speaker, who will take us through Parkinson's from a pandemic perspective.
Who has joined us from the Netherlands to talk about the Parkinson pandemic.
It is my absolute pleasure to talk to you today about the Parkinson pandemic, as we've come to name it, and obviously what we can do about it. My name is Bas Bloem. I'm a professor of neurology at the Radboud University Medical Center in Nijmegen in Holland, and delighted to see you today, if only remotely. These are all examples of people who recently came out as having Parkinson's disease, and the list goes on and on. You will recognize many of these familiar faces. Alan Alda was among the more recent people who also added his name to the list. For the fans, Ozzy Osbourne came out with Parkinson's disease, and I can already hear many of you thinking, these are all men, and this is true. The incidence of Parkinson's disease is higher in men than it is in women, but women are obviously not exempt. This is Linda Ronstadt, the well-known singer, who developed Parkinson's disease. And these are all examples of people that recently developed Parkinson's. And if you look in your own environment, you are probably aware of people in maybe your family or friends who also developed this disease. It's a fast-growing disease, and this knowledge is now substantiated by robust evidence. This is a paper published episodically in The Lancet or The Lancet Neurology by the Global Survey of Diseases, where they monitor time trends in the development of all sorts of neurological conditions. So this is not particularly focused on Parkinson's. It looks at developments for any neurological disease. And I've highlighted one key finding here, and I realize it's a busy slide, but will walk you through it. These are the data for just Parkinson's disease, stroke and Alzheimer's disease. Now, if you look at these data, all three become more prevalent as we grow older. So aging leads to an increase in all three of these neurological conditions. But in the right column, which is where you need to focus, they looked at age-corrected incidence of Parkinson's and the other two conditions. And after correction for aging, stroke is actually diminishing, which means that we're now better at preventing stroke. Alzheimer's remains more or less stable, which is explained by the fact that aging by itself is apparently responsible for the growth in Alzheimer's. And the only condition in this global survey of diseases that exploded even after correction for age was Parkinson's disease. So Ray Dorsey, my good friend and colleague from Rochester, and I published this paper in JAMA Neurology where we have called this the Parkinson Pandemic, and we coupled a call to action to it. And why we need action is not just because the numbers are rising, it is also because we can do something about it. Some people might argue, well, we've become better doctors, so this is why we diagnose Parkinson's more readily. But that's not the case. Multiple sclerosis has seen great advances in diagnostic testing with MRI, cerebrospinal fluid, but the incidence is hardly increasing despite aging of the population and despite advances in ancillary testing. We are still diagnosing Parkinson's disease more or less with our bare hands, listening to the story, looking at patients. Yet, despite the same techniques that we used 200 years ago, Parkinson's is exploding. So something else is happening, and we think that just like the pyramids, Parkinson's is at least in part a man-made disease. Not entirely, but at least in a large part. There are genes which explain a small proportion of Parkinson's. Typically those with an age at onset under 40 runs in the family, but for the large majority of Parkinson patients, genes are not the sole or the dominant role. And if you come to think of it, James Parkinson first described the disease in 1817 in London when they were having their industrial revolution, when air pollution was taking monstrous shapes, the notorious English fog. We've looked at industrial revolution in a recent paper, again with Ray Dorsey, that we published in Journal of Parkinson's Disease, and as a proxy for industrial revolution, we took the growth in gross national income shown here on the x-axis and the number of new people, the growth in PD on the y-axis. And you can see that there is a far from perfect, but there's somewhat of a trend in the relationship between it. And the one disease that really sticks out is China, where they are now having their industrial revolution and where Parkinson's disease has more or less exploded in the last decade. And this is a picture from China recently with monstrous air pollution. But it's not just air pollution. And by the way, a recent paper in JAMA Neurology confirmed again that air pollution by itself is one of the factors related to the growth in Parkinson's. But we are particularly concerned about pesticides like paraquat, which has been banned in the Netherlands in 2007 because it is severely toxic to the substantia nigra. It is still used today in the United States, which is, I think, outrageous, and in a number of other countries in the world. And remnants of paraquat are still persistent in our environment. Paraquat and other pesticides are very toxic to the substantia nigra, killing off dopaminergic neurons, causing Parkinsonian symptoms in rodents. In addition, and this is just one of various papers that have been published, there's a clear relationship between exposure to pesticides in humans and the risk of Parkinson's. This is born from a series of epidemiological studies, one of the more provocative ones being this paper by Alexis Elbaz in France, where they looked at a map of France and looked at the incidence of Parkinson's and overlapped it with rural areas, and in particular wine farming. And their conclusion was that particularly wine farming, but also other rural areas, were closely related to a higher incidence of Parkinson's. And the French have now gone so far as to recognize Parkinson's as an occupational hazard disease among wine farmers in their country. But the risk extends beyond just those who are exposed directly to pesticides. This is in Dutch. These are the famous Dutch tulip fields that attract hundreds of thousands of tourists each year. These tulips grow because the Dutch use pesticides on these tulip fields, and what surveys have now shown in the Netherlands, that in the houses in the immediate perimeter of the tulip fields, pesticides can be found in measurable concentrations on the kitchen table, on the floor, on the skin of the people living there, in diapers of babies and newborns, which is just incredible. So we talk about this in our book, Ending Parkinson's Disease. We talk about this pandemic. We discuss what needs to be done about it. And if you're interested, I hope that you'll find the book worth reading. The book is also a prescription for action. And that is literally depicted here in this cartoon. We emphasize that what is really needed is a pact to prevent Parkinson's disease by sanitizing our environment, by advocacy, raising awareness for this growth, also raising awareness for the need for more funding. It's a call for better care models and for better treatments to not only treat the symptoms, but hopefully one day slow down or even arrest progression of Parkinson's disease. And these are the other authors who have their own prescription package. So how should we deal with the pandemic? I always like to portray it as two parallel tracks. We need to do fundamental research into the mechanisms of brain death so that we can understand those better and ultimately tailor disease-modifying treatments to it. And then, of course, we need to improve our business to deliver better services to affected patients today. And thinking about the latter, I'm always inspired to look at outside healthcare. And for example, Steve Jobs, the famous guy who made Apple rich and large, always used to think very distinctly from the client's perspective. And he really wanted to know, what do my clients really like? And we followed in his footsteps by doing a surveying among patients. It's a method borrowed from industry. It's called the voice of the customer, which we used to arrive at the core needs of patients. It's a particular deep interviewing method. And what the patients told us in this survey is not that they wanted more attention. This patient is receiving a lot of attention by a number of nurses, a doctor, maybe a physiotherapist, but you can't say that we're taking this patient very seriously. What patients did tell us was a number of things. They wanted to participate in healthcare. They did not want to be treated as objects, but rather as subjects who contributed to their own health. My favorite new term for this is participatory health. They were craving for reliable information. They were in dire need of a single point of access, which I think could be a Parkinson nurse, who is readily available to answer questions, or to direct you to another professional who might answer the question better. And there are many professionals potentially involved in the care for people with Parkinson's. I had the immense privilege to write a seminar for The Lancet on Parkinson's disease. It came out as a 20-page review, and figure 8 is the star figure. It's shown here, and it portrays the multidisciplinary team where the patient is at the heart as the sun in the universe, and otherwise there are no stars in the universe, definitely not the doctor, but there are about 30 professional disciplines that orbit as planets around the sun, which is the patient and the family. In the Netherlands, we have organized this multidisciplinary care approach in a nationwide multidisciplinary network called Parkinson Net. We've trained about 3,500 professionals from 12 disciplines who we have trained according to guidelines and where we concentrate care among those dedicated experts. And we've looked at the cost effectiveness of this network approach in a series of papers, all published in high-end journals. And the bottom line, without going into detail, is that it leads to better care, fewer disease complications. There was even one of our studies showed a 50% reduction in hip fractures among Parkinson patients and a cost saving that amounted to 20 to 30 million euros each year for Parkinson's care alone in Holland. That was about a 5% cost reduction. So that is very promising and encouraging for the future. Another thing that patients keep telling us is, treat me as a person. This is the most compelling slide I'm showing today. It's Sarah Regara, who will be defending her PhD thesis at my hospital this year. She added up all the minutes of contact with her professionals, medical professionals, and today it was 24 hours per year, shown in red. The blue circles is when Sarah is the PhD student, the mother, the friend, the person. And I think this is such a powerful image that shows you that patients are so much more than just their disease. What patients really need is personalized precision medicine, care and treatments tailored according to their unique profile, but particularly to their wishes and needs. And what patients, of course, desperately hope is that there will soon be treatments that not only suppress the symptoms, but that may ultimately slow down the progression of Parkinson's. And the ultimate dream is the cure. Now, I think that in my lifetime, we will be witnessing a disease slowing. I think a cure is still a long way to go, but we should be fighting until we have at least that disease modification. And who knows, the cure may arrive at some point. I want to thank Herantis for inviting me for this presentation, and I look forward to interacting with you in more debates and about these important issues.
Thank you so much, Professor Bastiaan Bloem. Remember, guys, that you can ask questions in the live chat. And we will be right back with Henry Huttunen, the CSO at Herantis Pharma. Stay tuned.
Good afternoon. My name is Henry Huttunen, and today I'm going to, for the first time, show you data from our Cdnf development program. As you may have noticed, about a week ago, Herantis Pharma announced the selection of Cdnf preclinical candidate. This compound called HER96 is an engineered peptidomimetic, which is derived from the active site of Cdnf protein, and is also capable of penetrating the blood-brain barrier. So in this 25-minute presentation, I will first introduce to you the mechanism of action of Cdnf and explain how Cdnf is believed to protect the vulnerable dopamine neurons in the brains of Parkinson's patients. Then I will explain the rationale behind the Cdnf development program and show you data on the HER96, the preclinical candidate, and hopefully by end of the presentation you will agree that this is really an ideal candidate for future development. Today you will hear this word many times, proteostasis. Proteostasis is the process in all our cells that regulates the proteins, their synthesis, folding, and degradation. And this is very important for maintenance of the health of the proteome, the cell, and the organism itself. The lifelong maintenance of neuronal function and proteostasis is a major challenge. Majority of the neurons in our brains are born during the embryonic development period, and these very complex, both structurally and functionally complex cells need to be maintained in a functional and healthy state throughout our lives, up to 100 years or so. In several aging-related CNS diseases like Parkinson's and Alzheimer's, there's a very clear disruption of proteostasis, which results in accumulation of misfolded and aggregated proteins in the brain. And on the right-hand side here, you can see examples of a protein called alpha-synuclein, which is the major component of the Lewy bodies and Lewy neurites in the Parkinson's brain. The staining that you see in brown here is something that doesn't belong here. These brown structures here are Lewy bodies in the cell soma, and the thin Lewy neurites here are axons filled with aggregated alpha-synuclein protein. This results in cellular dysfunction and slow degeneration of both synapses and the cells themselves, and eventually development of clinical symptoms. There are several cellular functions, molecular networks, subcellular organelles that cooperate in order to maintain the proteostasis. And in Parkinson's, I wanted to highlight here 3 important cellular organelles that are important for proteostatic maintenance. These are the endoplasmic reticulum, shown in yellow here, this complex membranous organelle surrounding the nucleus, the mitochondria, and the lysosome. These 3 are in dynamic crosstalk all the time in order to make sure that both the synthesis in the ER and also the recycling of the proteins occurs in a balanced fashion. The endoplasmic reticulum, where Cdnf is located, interacts frequently with the mitochondria to maintain the cellular bioenergetics, but it also interacts with the lysosomes. Now, Cdnf is located here in the endoplasmic reticulum, where its function, its physiological function, is to regulate the ER stress sensing molecules and networks. Interestingly, if we remove the functional gene from animals, the functional Cdnf gene, what happens is that there's a loss of enteric dopamine neurons, and this coincides with the dysfunctional autophagy. Autophagy is a self-eating, a recycling process that's heavily dependent on lysosomal function. So this again illustrates the important crosstalk between the ER and the lysosome in maintenance of proteostasis. Now, in neurons, we know that endogenous Cdnf is located here in the endoplasmic reticulum. It regulates the cell ER stress sensing mechanisms, protects against alpha-synuclein aggregation and toxicity. There are also some other survival promoting effects, and also long-term transcriptional effects to support neuronal function and survival. These are all cell autonomous mechanisms. Importantly, Cdnf also has non-cell autonomous effects, and these are targeting the glial cells of the brain. More than half of the brain's cells are actually not neurons, but glial cells. And there's a very important cell type called microglia that comes from the same lineage of cells as the circulatory monocytes and macrophages. So these are immune cells. The cells are activated by many triggers, also including the aggregated proteins like alpha-synuclein. And when microglia is activated in a chronic fashion, they start secreting pro-inflammatory cytokines that, together with the neuronal processes, stress, chronic stress, and other processes, form a vicious cycle that eventually then results in degeneration. And Cdnf targets the neuronal processes, but also the microglial processes resulting in chronic inflammation. So back to the ER. Here, the target proteins the target mechanism of CDNF is the pathway called unfolded protein response pathway, UPR. CDNF interacts with several proteins involved with UPR signaling and modulates their activity. The three receptors of the UPR pathway are called IRE1, PERK, and ATF6. Their inactivating ligand for all three receptors is called GRP78, and CDNF modulates the function of this whole system via interactions with these four proteins. Importantly, the UPR can have two outcomes. In normal situation where there's an activation induction of ER stress, the aim of the UPR is to restore the cellular homeostasis, resolve the stress, and it works in the so-called adaptive mode. However, if the ER stress is very high or very long in duration, the UPR signaling can shift into chronic mode, and this supports pro-apoptotic signaling and results in cell death. And the function, the physiological function of CDNF is to balance these two pathways. There's now plenty of evidence from human diseases, from human genetics, from preclinical animal models, showing that the UPR pathway is very centrally involved in several pathologies of the brain, and it's a very well-established drug target for many diseases. Now, for many years at Herantis, we have had a goal to develop a CDNF-like protein that would not only have the neuroprotective effects of CDNF, but also would be capable of penetrating the blood-brain barrier and as such help to avoid the invasive intracranial delivery of CDNF. Here you can see a structure of the regulatory domain of the GRP78 protein located in the lumen of the ER, regulating the UPR signaling in complex with CDNF. So the blue here is the CDNF N-terminus, and the green is the C-terminus of CDNF. So we know that these proteins interact with each other, and we know that this green part of CDNF, the C-terminal part, binds to the UPR targets. Now, what's really interesting is that a few years ago, collaborators of ours at the University of Helsinki showed that this part of CDNF, if you make little fragments from this part of CDNF, they actually can penetrate the blood-brain barrier. Here you can see a brain section from a mouse, a healthy mouse with intact blood-brain barrier, injected subcutaneously with a fragment from this area of CDNF. And the red dots here are actually the peptide located in the substantia nigra, the key area of the brain involved in Parkinson's. So this part of CDNF is not only active site of the protein, but it also can be used for targeting the brain when smaller compounds are used. So in our lead optimization program, we identified very small parts, very small fragments of the CDNF protein that maintain these important properties. So here you can see the mother protein, 163 amino acid protein, complex folding in two different individual domains, and then examples of shorter fragments of ex-CDNF compounds, which actually maintain the neuroprotective property, the alpha-synuclein aggregation modulating property in vitro potency assay, and also in a cell-free binding assay shows that they still bind to the critical UPR targets. Now, native peptides, like the compounds shown here, are not very good drug candidates. So in our ex-CDNF lead optimization program, we used chemical engineering to take these short peptides and make them metabolically more stable and more drug-like molecules, resulting in peptidomimetic compounds. Here is shown the HER96, the preclinical candidate, compared to CDNF, the mother protein, in a simple in vitro neuroprotection assay. So here we use midbrain neurons in a culture dish, and we treat them with a toxin widely used to mimic Parkinson's disease in preclinical model. And what you can see is that both Cdnf and the Her96 very effectively protect the neurons, and at the same time on the right hand side here, they reduce the alpha-synuclein aggregation induced by the toxin. And what's interesting and very important here is that the blue curve here, the Her96, not only reaches higher level of neuroprotection than Cdnf, but it also reaches the high protection at lower concentration, suggesting that it's a highly potent compound, which is very important for any therapeutic that is systemically delivered and aimed to target disease processes in the brain. In this experiment, we have compared Cdnf and Her96, how they modulate the UPR target pathways. So the schematic on the right-hand side here shows a simplified version of how the UPR signals. So the IRE pathway activates certain targets. The PERK pathway has separate targets, and ATF6 has its own way of signaling. And what we're measuring here is phosphorylation of the IRE1 protein, shown in orange. Then we're showing a downstream target of PERK pathway, measuring nuclear localization of the ATF4 transcription factor, and also separately measuring the nuclear localization of the ATF6 transcription factor. The dark blue shows caspase 12 activation, which is known to be involved in the pro-apoptotic arm or mode of UPR signaling. And as you can see here, again, we used MPP plus toxin, which is killing some of the neurons. And this coincides with the elevation of all these UPR markers. And when we applied either Cdnf or Her96, we can see that there's a clear neuroprotective effect, but there's also a very clear and very similar modulation of the UPR signaling activity by both compounds, showing that the Her96 is really engaging with the same cellular targets and same pathways to protect the neurons as Cdnf is. This is a benchmarking experiment and study that we did with the Her96. Here we used 5 different known, widely used compounds for modulating single pathways of the unfolded protein response signaling system. And what's common with these compounds is that they have a bell-shaped dose response curve like Her96 does, both in terms of neuroprotection, but also in terms of alpha-synuclein aggregation. But the clear difference here is that all these the benchmarking compounds have a very narrow concentration, active concentration range, only spanning about 100-fold concentration range. This is, of course, a very important thing for human dosing, for example. Very importantly, here we can see the Her96 spans a significantly wider range of active concentrations, about 15,000-fold range of concentrations. So this is a very, very good property and clearly shows that the Her96 is superior over all tested benchmarking compounds that target the UPR pathway. So Her96 is a peptidomimetic compound, and it means that we have engineered a peptidic compound to become more stable metabolically, being able to resist cellular and also circulatory degradation mechanisms that exist in the human body. So here we compare a non-modified control peptide on the left-hand side to Her96, and we can see that the Her96 has very clearly superior stability over a non-modified control peptide, both in plasma and also in cultured hepatocytes. And on the right-hand side here, you can see a simple pharmacokinetic study from rats, where HER96 was administered subcutaneously, showing that there's about 20-minute plasma half-life. So this suggests that together with the fairly low plasma protein binding, that this compound has the primary route of elimination is via the kidney. Here's another pharmacokinetic study where we use dual microdialysis. We placed a probe in the jugular vein of a rat and also in the striatum of the rat. So simultaneously we collected samples from the blood and from the interstitial fluid of the brain parenchyma. And in this study, we very clearly showed that the brain to plasma ratio of HER96 compound is about 0.22. Also, the brain interstitial fluid half-life is about 1.3 hours. So these are good numbers for this type of compound, suggesting that this could be nicely dosed to humans also by subcutaneous route. And finally, we tested HER96 in a fairly novel model of synucleinopathy, of Parkinson's disease. And here we used aged black 6 mice that had been injected into the substantia nigra with alpha-synuclein oligomers. And together with this treatment, we started daily injections of an inhibitor, chemical inhibitor of an enzyme called GBA, glucocerebrosidase. This is an important lysosomal enzyme, also involved in some subtypes of Parkinson's disease. And together with the alpha-synuclein oligomer, presence of oligomers over the 4-week follow-up period, we could see that there's a very nice progressive development of pathology. So first of all, we have a clear induction of microglial activation, microgliosis. We have upregulation of several unfolded protein response pathway markers. And this coincides with the loss of dopamine neurons, as shown here on the green staining on the top panel. And the quantitation on the right-hand side here shows that this is truly a progressive model where both UPR and neuroinflammation are progressively upregulated. And in this study, we dosed HER96 3 times a week, starting from day 1, and followed the animals for 4 weeks. And at the 4-week time point, we then analyzed the brain for various markers. The left graph here shows the loss of dopamine neurons. The gray bar is the control animals. The red bar is the alpha-synuclein animals receiving vehicle. And then the 2 blue bars are 2 different dose levels of HER96. And as you can see, the higher dose level, non-optimal dose level, here has shown 46% protection of dopamine neurons. And on the right-hand graph, you can see that there's a nearly complete alleviation, eradication of alpha-synuclein aggregates in these animals. Looking at microgliosis, neuroinflammation, and the UPR activation, we can see that the higher dose of HER96 reached 84% reduction of microglial activation marker IBA1. And if we look at the phospho-IRE1 and ATF6 activation, we have 50 to 85% reduction of these important UPR markers. So this data clearly shows that we are engaging with the same targets that Cdnf is known to modulate both in vitro and in vivo, and also provides a preclinical proof of principle for HER96 efficacy in a relevant animal model. So in summary, we have developed a highly potent compound that mimics the activity of Cdnf. It is a very easy compound to synthesize. It's a well-soluble compound in aqueous buffers, easy to formulate. We have several backup compounds for the HER96 in the pipeline. We have excellent metabolic stability, low plasma protein binding, and we have determined that subcutaneous administration is an optimal route of administration for this compound. Very good brain to plasma ratio and robust disease-modifying effects in an alpha-synuclein-based model of Parkinson's. So the disease-modifying effects of CDNF are known to be based on three components: neuroprotection via modulation of the unfolded protein response pathway, reduced alpha-synuclein aggregation and toxicity, and reduced neuroinflammation. We have also now demonstrated that the ex-CDNF lead compound HER96 has the same effects as CDNF does. There's evidence of target engagement and therapeutic effects in a relevant alpha-synuclein-based animal model of Parkinson's. And we have optimized the compound to have improved BBB penetration, allowing systemic administration and a good metabolic stability and other pharmaceutical properties. And this, the size and the nature of the compound allows a simple manufacturing and reduces the cost. We have initiated the preclinical program of HER96 with an aim to start clinical studies in the first quarter of 2023. Thank you for your attention.
Thank you, Henry. And now I would like to introduce Dr. Magnus Sjögren, CMO at Herantis Pharma.
Hello. I will present the results from the first-in-human clinical trial in Parkinson's disease with CDNF. There were 17 individuals that received a surgically implanted drug delivery device, which you can see in the figures and the photograph to the right here on this slide. That was a drug delivery device that consisted of a port protruding through the skin, which you can see in the photograph to the down right. And to the upper right, you can see how catheters are extending beneath the skin and later on will extend down into the targeted brain area that is the putamen. This device enabled delivery of CDNF to the right area of the brain. And this was the first in human study assessing safety and tolerability first. It was done at 3 clinical trial centers, so Stockholm, Karolinska University Hospital with Per Svenningsson as primary investigator, Skåne University Hospital in Lund with Håkan Widner as investigator, and Helsinki University Hospital in Finland with Philip Schäferjans as investigator. Then we also had 2 PET centers, and I'll describe what PET is later on. Karolinska Institute PET Center with Andrea Verone as the scientist, and Juha Rinne at Turku PET Center in Finland. This study was co-sponsored by Renishaw Neurosolutions, who manufactured the drug delivery device, and also by a grant from the Horizon 2020 called the TREAT-ER. The study was designed in 2 active dosing phases and 1 safety stage. So the first stage, a main study, consisted of 6 months placebo-controlled double-blind randomized stage where 6 individuals received placebo or vehicle, 6 individuals received mid-dose CDNF, and 5 received a high dose of CDNF. Dose escalation was done from 120 micrograms via 400 microgram, which was the targeted mid-dose of CDNF, to 1200 micrograms, which was the targeted high dose of CDNF. After 6 months, the individuals exposed to placebo were switched to either mid-dose or high-dose CDNF in a randomized fashion. And now we are in the follow-up study where there's no exposure to CDNF, just safety assessment. The primary endpoints were safety and tolerability of intraputaminal CDNF, and we included some traditional efficacy outcomes as secondary measures. That's UPDRS for motor scores, PDQ-39, and CGI for global measures of Parkinson's disease. And then we included some exploratory measures, which I'll describe in more detail in the presentation. The clinical characteristics of the patients were that they were about 60 years of age in all dosing groups. They had an average disease duration of more than 10 years in all groups. All were Caucasian, mostly male, and they had an off time of about 5 hours per day. Safety was assessed at any time point during the study, so it could be reported by the patient, him or herself, spouses or family members or nurses or staff at the clinical trial site or by the investigator. And this was assessed together with other measures at each visit, of course. So we took blood and urine samples for assessing laboratory safety, as well as antibodies towards Cdnf. And physical examinations, including electrocardiograms, were done at each visit, and also neurological examinations. And then we took brain scans using magnetic resonance imaging at each third month to assess whether or not any structural changes had occurred. And we also used questionnaires for cognition, for mood changes, and for impulsive compulsive signs that may develop during the disease. Altogether, there were 68 drug-related, that is CDNF-related adverse events reporting during the 12-month treatment period. Most of them were mild to moderate and mostly transient. They were divided into these 5 main categories. So, for example, central nervous system disorders, which included then dyskinesias and headache, but also small air bubbles, fluid pockets near the catheter tips. These fluid pockets were assessed by the investigators, by external experts, including surgeons who had worked with the drug delivery device in the past, and also by an external independent safety committee as harmless, and the expectation was that it would resolve spontaneously. We also saw some impulse control disorder in 2 individuals, feeling of cold, fever, tiredness, some nausea, and some weight loss reported. But altogether, it was evaluated and assessed as Cdnf is safe and tolerated over 12 months intraperitoneal infusions. The highest cumulative dose was 10.6 milligrams of Cdnf. And the majority of the adverse events were mild to moderate and transient. And some of the adverse events, like headache, dyskinesia, impulse control disorder, nausea, may actually be due to an increased dopamine effect that could be a drug defect. And the serious adverse events reported were all related to surgery, to the drug delivery device, or the infusions, and not to Cdnf in itself. There were similar safety profiles in the main and extension study and between placebo, mid, and high dose. No differences in safety profiles. And there were no dose-limiting toxic effects observed. So we conclude 12 months of Cdnf is safe and tolerable. And we developed routines to improve the safety of use of the device during the study proceedings. Now, as mentioned, this is a safety and tolerability study, so we did not expect to see any efficacy issues or efficacy signals. And this is due to that the patients in this study were at a rather advanced stage of the disease. They had a disease duration of more than 10 years on an average. And when you look at their brains, like this study by Cordover, actually most of their dopaminergic neurons are already degenerated and lost. So it was for ethical reasons, since Cdnf have never been exposed to individuals before, and this drug delivery device was also used for the second time only, that we included individuals with a rather advanced stage of Parkinson's disease, thereby not expecting to see any strong signals on efficacy. We did, however, look at the secondary outcome measures, and this UPDRS graph here shows that there were no changes between any of the dosing groups, that is placebo, mid, or high dose. In terms of statistical analysis, no differences between the dosing groups. However, there was in general an improvement in motor scores to a 1.4 to 2.6 on the UPDRS scale over 12 months. As said, this was a small study, not powered to measure efficacy. And now, if we go into the exploratory measures, we used the DAT-PET, which is then a brain imaging method to reveal the number of viable dopaminergic neurons in the brains of these individuals. And this was assessed at baseline, 6 and 12 months, and 19 months. We also looked at the concentration in blood and spinal fluid of Cdnf, and we looked at pathogenic proteins such as alpha-synuclein in the spinal fluid, and we looked at all the proteins in the spinal fluid. And thereafter also we did a genotyping in some individuals, and also we looked at an objective measure for measuring motor movements called Parkinson's kinetograph, which is wrist worn, and it was worn a week before each dosing. Let's start with that, the kinetograph. So as you can see on this graph, there was a clear difference between placebo, which is gray, to blue, and green, which is high and mid dose respectively, on each time point measured. So that is 1, 2, 3, 4, 5, and 6 months during the placebo-controlled stage. So dosing with Cdnf was objectively, or was in better than placebo, using an objective measure for motor movements. And looking at the dopaminergic viable neurons in the brains of these individuals, you may expect a change of 6 to 13% in the number of viable dopaminergic neurons over a 12 months time period in Parkinson's disease, while almost no change in healthy individuals. And as you can see down to the left here, this was a placebo-exposed individual, and you can see the area of yellow and red in the picture, which decreases from baseline to 6 months, indicating a decrease in the number of viable dopaminergic neurons. And down to the right, you see the opposite, an individual exposed to CDNF, where the area is increased. Now, overall, there were no changes from baseline to 6 months to 12 months in any of the dosing groups that we could regard as clinically significant. However, there was a significant difference, an improvement in the mid-dose group at one time point in terms of the number of viable dopaminergic neurons. But as mentioned before, this study was not powered for measuring efficacy, so this is a finding which you may need to interpret with caution. We looked at also the alpha-synuclein proteins, the pathogenic protein that accumulates in Lewy bodies, the pathological hallmarks of Parkinson's disease. And we compared baseline to 6 months to 12 months, and there were no differences in any of the alpha-synuclein measures we used. For the genetic data, there were 13 that consented to genotyping, and 4 of them had carried a genetic risk variant for Parkinson's disease. So one individual had a LRRK2 mutation, G2019S, which is pathogenic. Another individual had a similar type of LRRK2 mutation, but this variant is of uncertain significance. Then we had one individual with a GBA mutation, which is the most common and which is pathogenic, and one with a VPS mutation, which is of certain significance. As mentioned, we also looked at the proteome, all the proteins in the spinal fluid, and we did this by first a discovery study, which then compared the individuals in our study at baseline to an external group of healthy volunteers. And from that, we identified, together with studying the literature, a set of 50 targeted protein that are relevant to Parkinson's disease and the mode of action of CDNF. And these included then immune response biomarkers, oxidative stress, autophagy and lysosomal function, ubiquitin, proteasome system, mitochondrial function, and axonal and synaptic adhesion and function, and so on. And looking at this slide here, you will see the differences at each time point for each individual. And I'll walk you through this rather complex slide. So if you look at between the header and the graph, you see it says placebo. That's the 5 individuals that first received placebo and then were switched to active dosing. 0.4 milligram CDNF is the mid-dose group, and 1.2 milligram CDNF is the high-dosing group. And each of these columns, that is 2 columns, where 5 are marked with blue frames, represent each individual included in this trial. And down below the heat maps, the colored heat maps, you can see whether or not they received placebo and then switched to mid-dose or high dose after 6 months. To the left of the heat map, you can see the functional classes that these proteins represent. For example, immune response, oxidative stress, lysosomal function, autophagy, trafficking of proteins, and so on and so forth. Now, you can see the first 6 months and then the latter 6 months displayed here for each individual. And the blue frames mark 5 responders. And response is an increasing red color intensity, which reflects an increased concentration of the relevant proteins for each functional class. Whereas blue, or increasing intensity of blue, denotes a decrease in the concentration. There were 3 that were of these individuals that were regarded as responders out of the 5 that were associated also with a clinical response and or a response in DAT-PET, that is the number of viable dopaminergic neurons. So I'm going to walk you through these 3 cases. The first case was an individual with a LRRK2 mutation, 62 years of age, more than 10 years disease duration, who received placebo, as you can see in the upper mid graph here, in the UPDRS and lead. It says first placebo for 6 months, and there basically no change. But switching to Cdnf, he experienced a 12-point improvement in UPDRS, which was associated with a stabilization of the number of dopaminergic neurons viable, which you can see to the graph down in the middle, and associated increase when switching to Cdnf in the relevant proteins in the spinal fluid. So this may indicate that LRRK2 mutation G2019S is a sensitive genetic risk variant for the mode of action of Cdnf. This second case is a 54-year-old female with 9 years disease duration, and which received mid-dose, both for the main study and the extension study, and experienced a 10-point improvement in motor scores, while the DAT-PET binding, the number of viable dopaminergic neurons, there was basically no change. However, it was a clear increase all the way through the 6 and 12 months in the relevant targeted proteins. And the last case here, a 54-year-old male with 10 years of duration who also received mid-doses all the way through. You can see on the UPDRS and lead graph that there was no change from baseline in motor scores, no improvement, no change, just a stabilization. Well, but if you look at the DAT-PET, the number of viable dopaminergic neurons down in the graph in the middle, where it says DAT-PET, you can see that there is a vast, huge improvement in the DAT-PET binding. We only had spinal fluid for 6 months, so you can see there is a corresponding increase in the targeted protein in this individual. Now, we looked at the proteins that increased the most during the exposure to Cdnf and identified 20 proteins that sort of moved in a concerted fashion, increased throughout Cdnf exposure. And what function they reflected was interesting, since it reflected the mode of action of Cdnf, namely lysosomal function or autophagy, immune response or neuronal cell adhesion, and synapse assembly. So the key takeaways and summary of this trial. This was a successful trial in that it met the primary outcome measures of safety and tolerability of 12 months of intraepithelial exposure to Cdnf. And we did explore some measures and also followed secondary outcome measures. And we find a promising biological response to Cdnf. This may be a result thereby of a Cdnf treatment effect, and some biomarkers are identified for future use in further development of this compound. In addition, we may have identified a group which is maybe specifically sensitive to Cdnf exposure, and that is the LRRK2 genetic risk variant G2019S in Parkinson's disease. And we will need to expose individuals that are at an earlier stage of the disease, who have more viable dopaminergic neurons left, in order to demonstrate the true potential of CDNF, that is disease modification. And we have also decided to switch the route of administration from surgically implanted device to intranasal spray and or subcutaneous exposure. And that we are developing as we speak right now.
Thank you very much, Magnus. And now I would like to invite Russell Patton. He is the Director General at the European Parkinson Disease Association. Over to you, Russell.
Well, thank you very much for inviting me. My name is Russell Patton. I'm the Director General of the EPDA. Delighted to be with you today on this webinar. I'm going to say a few words, obviously, about the organization that I'm representing. A few little words about the disease to really home in on, I think, one of our key messages. And then it's about very much our relationship, and in fact, very close synergies with the treatment industry. I'll then talk about the role of innovation, our view on genetics and genetic research, and in particular about the patient experience on clinical trials. And finally, of course, what would be our recommendations to scientists and clinicians who are working with patients, and in the case of the Parkinson's community, working with PWPs. Okay, so let me get into the meat of my presentation. So first of all, a few words about the European Parkinson's Disease Association. We are an umbrella organization, and we pan 28 member organizations across 20 plus European countries, and we go further afield into Ukraine, into Israel. Our members are, normally speaking, national associations or national groupings of Parkinson's people, but increasingly we also have organizations that just bring together a number of people with Parkinson's around a specific topic or field or interest, such as sport or music or dance, and that we see as a changing landscape. We work with many different stakeholders. Obviously, at the heart is people with Parkinson's, what we call PWPs, and increasingly carers. And I'm particularly keen to put the carers very much at the heart, together with PWPs, because at the end of the day, they are supporting the PWPs in their day-to-day activities. We, of course, work very closely with decision makers, so the political elite class in Brussels, and that means the European institutions, the European Commission, the Council of Ministers and their permanent representations, and the European Parliament. We work a lot with, of course, researchers, HCPs, and last but not least, the treatment industry, who we see as a very close stakeholder who ultimately will find, of course, better treatments and ultimately the cure. The EPDA strategic goals today are very much based on 4 key pillars. The first is to ensure that we help to get the right information at the right time to people with Parkinson's and their carers. Secondly is to strive for healthcare systems where people with Parkinson's can receive early and appropriate treatment, as well as individualized care. We continually need to raise the awareness of the complexity of Parkinson's and the impact it has on people's quality of life. And fourthly and lastly is to support the global Parkinson's community in the search for a cure. I'm not going to spend any time on the actual disease because I know some of the previous presenters have gone through it, but I think one key message is to say this is no longer a disease of the old, it's also a disease of the young, what we call the young onset, the YOPD community below 50, and there are now more and more cases of people even in their 20s, even children who are now have got the disease. And at least 10% of people with Parkinson's are under the age of 50, and we expect perhaps that percentage is much higher than what we actually have at the moment. As you all know, it's a dreadful disease. I'm not going to go into the details. Suffice to say that it's still not very well recognized. And I think there's always the adage of the person walking down the street who looks drunk. No, they're not. They are PWP. So in terms of our relationship as a patient organization with the treatment industry, myself, my members, my board, my annual general meeting members, we fully believe in working closely with the treatment industry. It needs, of course, to be a win-win, and there always needs to be a key bottom line that whatever we do with the treatment industry has a direct benefit for the PWPs. We also believe that cooperating with the treatment industry is the way forward. And what we are finding over the last few years is how patients themselves, people with Parkinson's in our disease area, are actually wanting to collaborate, to cooperate, to work, and to know more about what the industry, the treatment industry is doing. I was asked to talk a bit about innovation, and I guess innovation subtly, but is very much within the 4 core strategic pillars of the EPDA. Our number 2 objective is to help strive for healthcare systems where people with Parkinson's receive appropriate treatment and individualized care. And our 4th goal is the goal to search for a cure. So innovation is very much embedded in those 2 pillars. We want more innovation, and as a group supporting WPs, we need to see more innovation, and hence why we are proactive in supporting the treatment industry, including, of course, the latest Herantis clinical studies. I think we do appreciate the situation. There's a lack of innovation in disease-modifying treatments, albeit that Herantis' Cdnf is a very exciting prospect. And indeed, this is where we need to see change. But we also take note of the fantastic work that's being carried around the world around the development of increasingly effective symptomatic treatments. And there is no doubt we are in a much better position today than we were 10, 30, 40 years ago. But if we are honest and brutally and even a little cynically, we've still got a very, very long way to go. Without a shadow of a doubt, managing the symptoms of Parkinson's has hugely increased, as I explained, and it's increasingly fulfilling our number 2 strategic goal, and hence our call to develop to decision makers to not only raise the awareness of the disease, but so that they can appreciate that they need to support us in supporting further R&D. Genetics is becoming a hot topic. It's also a sensitive topic, and it's actually a topic that's really rising up our strategic pillars or our attention. We're always, as the EPDA, focused on where can we help most, and supporting better treatments and the search for cure are definitely such areas. But we're only slowly embarking on looking at how we can support studying the causes of Parkinson's. But I would say actually in the last 12 months, our members have asked us to look at increasingly environmental and chemical problems, and last but not least, genetic causes, as PWPs themselves are wanting to know more about the impact of these. On genetic testing, the EPDA is helping to lead the World Parkinson's Coalition Working Group on YOPD, where we will be focusing on this sensitive subject as it has a clear interest to this younger community of PWPs, the YOPD community. The EPDA's approach to better treatment, but also prevention and early diagnosis, is leading us to look at the genetic links to Parkinson's growth and how scientists hope to develop better and more reliable tests to identify those who might be at risk. This could help with the early diagnosis even before symptoms appear. We are convinced that assessment of genetic risks and early diagnosis will become increasingly important in the treatment, or to foster the treatment to start sooner rather than later. In terms of research, this has become one of our strategic priorities. Whilst we are not a research association, we are a patient-led organization, research is very much part and the soul of what we're doing. And because of this, we have recently formed a research interest group whose aims are threefold. First is very much identifying and address the barriers and solutions to engaging with research. Raise awareness, improve access to clinical trials, and thirdly is explore other opportunities to support Parkinson's research. Through our own member surveys, focus groups, monthly research meetings, we are gathering intelligence every day on how people living with Parkinson's wanting and are participating in clinical trials. If I was to give you some information in terms of the patient experience in clinical trials, I think these are what we would call some of the barriers that have been given to us, and in no particular order, but these I would say are the key ones. And many of these tend to be very common sense or very obvious. But perhaps because of time and speed and resources, we sometimes forget how these basic elements can become barriers in themselves. Firstly is accessibility and travel and transportation. I've heard patients having to travel 6-7 hours each way just to be part of a clinical trial process. Lack of information. Patients need a lot of information. The fear, of course, of side effects and whether going through clinical trials would change in a way their medication routine. The lack of consultation and support from HCPs, the inconvenient timing, poor communication, that's before, during, and after, and then perhaps generally a lack of collaboration between research centres and institutions. And interestingly, in one of our surveys, a third of respondents did not feel that they had the tools or resources available in their own mother tongue or their own language. So to end my presentation, I wanted to come up with what we think are our recommendations to scientists and clinicians. And please take them as, you know, we know you're doing a fantastic job. We want to see more innovation. And by that, I mean, we want to see more collaboration between the treatment industry and people with Parkinson's. So addressing some of those barriers from the EPDA's point of focus, it would be more training and education, better dissemination of information and clinical trial aggregator tools, and sharing of health data. And our own recommendations to scientists working with patients to find new treatments, new medicines, is first and foremost respect and understanding. Consider the needs of the patients, but also their carers who will be closely associated to what they will be doing. Trust and transparency. It takes time. We appreciate that. Realistic expectations, flexibility. Not always easy to work with patients. You need to adapt to them. They can't so easily adapt to you. Input, take and understand the input from the PWPs and indeed from the patient organizations themselves. And last but not least, I think it is communication. What are the aims? What are the clear objectives? Provide them with regular updates, review and evaluate. And remember, tailor your communication. Use a patient-first language. They're not scientists. They're people like me, we're generalists, and we just want to be able to understand the whole process of clinical trials. And so as scientists, as clinicians, you're going to have to adjust your language to fit to them and to their understanding. So thank you very much for your attention. I hope this presentation, these slides were informative and gave you an insight to the EPDA and what we do. And naturally, if you have any questions, please do not hesitate to reach out to me in the EPDA, and I will be only too pleased to answer your questions. And on that note, I wish every participant a good webinar and safe times ahead. Thank you very much.
And thank you very much, Russell, for an interesting presentation. And last but not least, we have Håkan Widner. He's a neurologist at Lund University Hospital, and he's here to give you his presentation.
Okay, thank you very much. My name is Håkan Widner. I'm a neurologist at Lund University Hospital in the south of Sweden. I'm to give a short introduction into how we look at Parkinson's disease and the cause of the disease, and some novel aspects of how we hope to be able to repair and change the course of this condition. So I'd like to give a little bit of a background into this work on how to restore the brain in neurodegenerative disorders. And just to give a little bit of the terminology, restorative treatments is really attempting to regain lost functions. It's slightly different from disease modification, where we try to limit a progressive course. That may actually lead to improvement, and there is sort of an overlap in terms of this, but we try to use the terms disease modification, which is a little bit more limited. We should also be aware of what we are working with. Everyone who sees a patient with tremor and walking slowly in the street has about 50% chance of saying that that is Parkinson's disease. In fact, there are many other causes. So we divide this is the diagnostic map. So we divide the symptoms of Parkinson's, the bradykinesia, rigidity, tremor, into the primary and the secondary forms. And they are a little bit more prominent where there's a primary form, and this is an active disease process, a neurodegenerative process. But of these, about 50% are what we call atypical, and it's actually different disease processes. Multiple system atrophy involves alpha-synuclein, but these disorders involve another protein called tau. And what we're really interested in this context here is Parkinson's disease, and that's a split between hereditary forms and where we really don't know the cause, idiopathic. When it comes to the pathophysiological mechanism in Parkinson's, it's quite a lot of known. It really involves the power steering for all our movements and anything that is involved in sort of planning movements, planning ahead, and so on. And the machinery involves 1 million nerve cells of the 100 million nerve cells that we have in the brain. And these nerve cells are located in the black substance, and they project about 5 centimeters long fibers into an area called the putamen and the caudate. Each nerve cell ends up with a synapse, and in there, dopamine is stored. And that is a neurotransmitter that is important for movements. We have something like 20 billion synapses normally after we are born. And for some reason in Parkinson's disease, these nerve fibers starts to degenerate, and that results into loss of dopamine. This is the normal content that we imaged with a radioactive substance. And here is a patient with a few years Parkinson's disease, and it's a more pronounced loss on this side. And this results into the symptoms of bradykinesia, slowness of movements, muscular rigidity, stiffness, and shaking or tremor. Here is a patient with a few years of Parkinson's disease, 7 or 8 years, not perfectly medicated at this moment. But this really demonstrates the slowness and the rigidity. It's leaning a little bit towards the left, taking small steps. And this is one of the hallmarks of Parkinson's disease. And the other one is this type of resting tremor. So it's when you're not really doing anything, there is a tremor. And when you grab or and reach for a glass, there is tremor that is going down, and it comes back when there is no activity anymore. This is caused by a disease process that resides in the dopamine nerve cells. And this is then the black substance that fades away over the years. There's a slow degeneration, so we lose cells. And we know that the process involves nerve fibers that aggregate a protein called alpha-synuclein. And that is transported from the brain region back to the nerve cells in the substantia nigra and is lumped into what we call Lewy inclusion body. And that's the hallmark of Parkinson's disease. So we are actually looking at Parkinson's disease when we see this. This can only be done in autopsy after death. So we have no way of seeing this at the moment. But we can image and see quite a lot of what Parkinson's disease can do. And this is the network of how nerve cells work together. The brain is mainly electrical and sends electrical information between various regions of the brain. And at the end of each nerve cell, there is a chemical intermission with a substance, a neurotransmitter that translates the electrical signal into various functions through a chemical step. And the most important one is dopamine. It's actually discovered here in Lund for more than 60 years ago by Arvid Carlsson and his co-workers. This is the dopamine content and nerve cells ending up and producing dopamine and releasing dopamine when there is an electrical signal coming. This is an image of how dense the nerve cells are in the brain in this region of the putamen up here. And the enormously rich and complex nerve contacts. And these nerve contacts form bundles and relay information that is important for us to move in a proper way. Apart from dopamine in Parkinson's disease, other neurotransmitters are also affected, norepinephrine and others. And this network here is really the basis for some of the understanding and new treatments that we are working with in Parkinson's disease, including deep brain stimulation apart from medications. The main problem in Parkinson's disease is the disease progression. If we could freeze and hold the disorder, we have pretty good treatments at the moment, but we have at the moment nothing that really controls the actual disease process. And this is possible to measure. So this is a patient that's undergone repeated scans and one can that measures dopamine content. And one can see over a course of 5 years that the symptoms increase. This is rated by a rating scale. And we can also see that there's about a 10% loss of this signal. That's not really equal to loss of 10% of cells, but it is the dopamine content that goes down. And this is sort of a schematic cause of Parkinson's disease. We know that Parkinson's disease starts several years ahead of the onset of visible symptoms in terms of motor symptoms. And then there is a slow but clear progression of the various symptoms. And apart from the motor symptoms, there is also a huge array of what we call non-motor symptoms, including apathy, fatigue, sleep pattern that is affected, and orthostatic hypertension and so on. When it comes to treatment, it is symptomatic still after all these years. We have very good treatments, and for a Parkinsonian patient with the true Parkinson's disease, there will be medications that work throughout. It's a myth that medication stops working after a few years. But there is quite a lot of sort of inherent problems with the medications. So we usually try to aim for a combination of levodopa and dopamine agonists, MOB inhibitors, COMT inhibitors that are enzyme inhibitors to modify the levodopa and the dopamine content. And we try to avoid very high or very low doses. And this needs to be adjusted on a very regular basis throughout the disease course. There are many handbooks and recommendations of this, for example, from the Movement Disorders Society, and there are also national guidelines of various kinds. When it comes to the cause and of Parkinson's disease, it is a multifactorial, and some also say that there is not one Parkinson's disease, but several. And then we don't regard it as atypical, but it's various forms of Parkinson's disease. And one can split this into various conditions, such as tremor dominant, which tends to be younger, slightly less aggressive disorder. And then we have the ones that goes without prominent tremor, about 25%, that has perhaps a little bit more aggressive course with prominent falls that develop fairly early. There are about 10% that has a genetic factor that is really causing disease, the monogenetic forms. There are mutations, the alpha-synuclein, for example. And then we have risk genes, or susceptibility genes, and about 90 of those are known and affect Parkinson's disease and its cause. But they are not really causative. So it's quite important to distinguish between the ones that actually cause the disease and the ones that modify it. And then there are environmental factors. The most important one is age. This is highly age dependent. And then there are several other factors. There's been a lot of discussions if repeated head trauma is a risk factor. It's been in all textbooks throughout the years, but when in studies here in Denmark and Sweden has evaluated that, it is possibly so that it is actually the onset of early Parkinson's, the prodromal form, that leads to repeated falls and head injuries in a group of patients. So it is not really quite clear. And the one professional boxer, for example, that develop secondary Parkinson's has a different cause. There are some known protective factors. The strongest one is repeated use of a non-steroidal anti-inflammatory drug, ibuprofen, and smoking and coffee that can reduce the risk quite extensively. When it comes to the sort of mechanisms of the disease, it is still not really known, and it's become exceedingly complex, and I'm not going to go into very prominent details in these. But I could say that when I started out in the late 80s, the sporadic and toxic factors, such as a potent toxin called MPTP, was all in the range. And this involved the metabolism with the loss of complex 1 in the mitochondria, generation of free radicals, And quite a lot of studies in trying to interfere with the disease process has been done. Unfortunately, not really effective. But the findings are there, that there is a complex 1-energy deficiency in Parkinson's disease, and that's been repeated over and over again. With the event of genetic forms, various factors in the metabolism, energy production, but also the metabolism of one of these proteins, alpha-synuclein, has been a focus for understanding Parkinson's disease and how to interfere with this. This slide is from 2008. All the key players are there. We add in genes, for example, and one has developed this over the time. And this is roughly the same image here with alpha-synuclein as a main culprit in Parkinson's disease, driving Parkinson's disease and disease degeneration of the nerve cells. And all the various components with gene factors and inflammation, toxic effects of oxidative stress, and lack of support is involved in the process. And one of these quite interesting aspects that we can mimic in the laboratory and to some extent in animal models is with alpha-synuclein. Here are some of the key players. Alpha-synuclein is actually degraded and then can be involved in the disease process. Indeed, this has been translated into attempts of affecting the disease process by complexing alpha-synuclein with antibodies, monoclonal antibodies. And this is ongoing, an attempt of trying to modify the disease cause. Here we can see cause where alpha-synuclein is sequestered by these antibodies and it's bound. It's not translated into a clinical effect yet, but treatments are ongoing. Another aspect of trying to repair the brain is something that we are very much involved in here in Lund, is trying to replace nerve cells. And we are currently working with dopamine nerve cells produced from stem cells and for a transplantation approach. That has been done with fetal tissue, and one can see in those conditions that alpha-synuclein is actually aggregated in the novel cause or novel cells that have been transplanted that could mimic that there is a disease process going on in the brain of Parkinsonian patients. And then I would like to go over to some of the focus of this seminar and the interesting compound Cdnf that we have tried in a clinical first-in-man trial. And Cdnf is really pertinent for the disease processes in Parkinson's disease. It has many actions, modes of actions. It restores the normal proteostasis, the turnover of proteins, for example, alpha-synuclein, that is really involved, we think, in the disease processes in Parkinson's disease. And by doing that, it reduces the neuroinflammation that is also an important component in driving Parkinson's disease. Cdnf can protect neurons, and especially vulnerable neurons under an attack of a disease process, where Cdnf has shown protective effects, and it normalizes the alpha-synuclein and reduce the aggregation. In this clinical trial, it has been shown that Cdnf, when we administer it to patients, it's safe. It's no major safety concerns, and it can modulate CNF proteomics and as biomarkers for the disease process. And in one patient, for example, here in Lund, happens to carry a genetic marker, LARC2, for Parkinson's disease. We have seen a very good clinical response that's been long-lasting. It has reduced the fluctuations, reduced the need for medication, and indicating that Cdnf can have important functions in modulating Parkinson's disease. So we are really approaching something that can modify Parkinson's disease. The underlying biologies are very complex, and there could be both direct and indirect effects, symptomatic and disease-modifying ones. And it really underscores the need for a very careful clinical trial design to tease out the possible mechanisms. This takes time, and I'm afraid that patients need to be patient, but we are in the process of making progress towards a disease-modifying treatment, I hope. Well, this concludes my presentation. I hope it's not been too complicated, but we are quite confident that will be some very interesting and exciting news around Parkinson's disease in the future.
Thank you very much for everyone for joining today, and thank you to the speakers for giving such interesting presentations. I will now hand over to the CEO again, Craig Cook, for a closing statement before the Q&A session.
Thank you, Professor Wittner, for that most useful and insightful presentation. So we have covered off a lot of relevant ground today, including Parkinson's disease generally, as well as CDNF and ex-CDNF specifically. Our external speakers have spoken about Parkinson's from a population and pandemic perspective. They've spoken from a patient perspective, as well as from a physician perspective. And our internal speakers have focused on CDNF and ex-CDNF and the key aspects of those treatments as they relate to Parkinson's disease, including biomarker data, clinical data, imaging data, as well as genetics, as well as the correlation between those key important areas of Parkinson's treatment. This has been a lot of information to communicate. So in summary, I would like to highlight some of the key points to remember from the presentation. So key point number one is proteostasis and the fact that CDNF and ex-CDNF act powerfully on this key system of the body to restore proteostasis and correct the pathology of Parkinson's disease and other neurodegenerative diseases. So that's key point number one. Key point number two are the proteostasis biomarkers, where we see clear evidence for both CDNF and ex-CDNF from these biomarkers that the proteostasis is restored in response to CDNF and ex-CDNF. That's key point number two. Key point number three is disease biomarkers, where we see these markers of disease in humans respond to CDNF treatment. We also see that the response in these disease biomarkers correlates with motor score improvements as well as imaging improvements in these patients. That's key point number three. Key point number four, CDNF is safe in humans, as established very successfully in the phase 1 study completed last year. And key point number five is the convincing ability of ex-CDNF to penetrate and cross the blood-brain barrier, as well as protect neurons in the brain. These are notoriously difficult things to achieve, but ex-CDNF has achieved exactly that. So that is a strong foundation to build on as we move forward. And there's an intensive and comprehensive program planned for the future, as we see in this slide. Cdnf is a clinical stage asset. It's at phase 1. We have the alternative routes of administration that we're currently developing, nose to brain and subcutaneous. And then for Cdnf, we've just selected the lead compound or the candidate that we're going to take into further clinical development. And allied with the activities on the left-hand side of the slide, on the right-hand side of the slide, we see a rich planned news flow for the next 12 to 18 months. So in conclusion, and the key takeaways from today's session regard our compelling science, our exciting data, and the significant opportunity at hand. Compelling science as it relates to evidence of target engagement with proteostasis, exciting data as it relates to Cdnf from a clinical perspective, a biomarker perspective, an imaging perspective, as well as a genetic perspective. Exciting data as it relates to Cdnf, where the re-engineering, the chemical re-engineering of Cdnf has been very successful, as evidenced by the ability to cross the blood-brain barrier and the highly potent nature of the effects of Cdnf. There's a significant opportunity at hand, 10 million patients worldwide. The total neurodegenerative market is predicted to be upwards of 60 billion by 2026. And for Herantis and our assets in Parkinson's disease specifically, that equates to an $8 billion opportunity. The news flow over the next 12 to 18 months is planned to be robust and rich. And finally, we have a highly skilled team driving all of this. We know what is needed. We know how to do it. We have a strong track record. And we have some of the brightest brains I have ever worked with in my career involved and driving these programs. I trust that you have found the Herantis R&D Day enlightening to Parkinson's disease generally, as well as to the prospects of Cdnf and Cdnf as it relates to the treatment of Parkinson's disease. I thank you for joining. I thank our external speakers once again for participating, and we will now take a Q&A together with the team.
All right, now it's time for the Q&A session, and we have received a couple of questions beforehand. And the first question is for you, Craig. Someone wants to know if you could please elaborate somewhat on the interplay between Cdnf and Cdnf. What is the reason behind having both in development?
Sure, Kaiser. So Cdnf is a clinical stage asset. It's a biological protein. It's completed phase 1. Cdnf, on the other hand, is a preclinical, earlier stage asset, chemically engineered version of the parent Cdnf. And right now, the respective merits of each program merit that we pursue both programs. There's a lot of knowledge transfer between the programs, and we'll continue to evaluate the respective programs as we proceed through the different development stages. But certainly now the rationale and the scientific merits for pursuing both programs is strong.
Okay, so you will continue to develop Cdnf as well?
Mm-hmm. Yes.
The next question is for Magnus. It's from a Red Eye analyst, and it says, you mentioned that you saw early indications of higher response to Cdnf in patients with specific genetic mutations, such as LRRK2 mutation. How will this finding impact upcoming studies? Could a confirmation of this finding affect target population, pricing, etc.?
Yeah, thanks, Kajsa and Red Eye. So It is a very interesting finding of this clinically extended and clinically significant response in an individual with the Parkinson's disease pathogenic gene variant called the LRRK2 or LRRK2 mutation. So it may indeed impact our development. We are investigating and exploring this route of clinical development into this potentially specifically sensitive subgroup of individuals with Parkinson's disease. It may affect our clinical development. It may affect a lot of things, but it's for us at the stage where we are in right now, very interesting finding. And that's what a lot of companies are exploring and also pursuing. Do we find a sensitive subgroup of individuals with Parkinson's disease? Then that's the shortest way to the market.
Okay, then let's talk about the mechanism of action. With the proteostasis, how does Herantis compare with competitors?
Maybe I can briefly answer that, and then Henry can add more detail. Well, proteostasis comprises three key systems. It's the production of the proteins, the folding of the proteins into their active structure, and the degradation of the proteins, or sort of quality control to eliminate toxic proteins or too many proteins. And then in addition to that, there's an inflammatory component outside of proteostasis that also contributes to collateral damage, we can call it that. And many of the other companies are focusing on the degradation elimination part of the equation. For Herantis, we cover that, but we also cover the other four components, the other three components, the total four components that I mentioned. So we go way beyond what the other competitors are doing, who are focusing just on alpha-synuclein. They're doing great work, but equally we're doing great work on further components of proteostasis. Henry, you may want to add some more detail to that.
Yeah, thanks, Craig. I think it's also important to, if we just specifically talk about the stress responsive pathways, to keep in mind that traditionally the unfolded protein response pathway has been approached with selective inhibitors of single pathways, and this has turned out to be not really an optimal way. And while CDNF binds to multiple unfolded protein response pathway targets and modulates the activity of the whole UPR system, keep it in a mode that protects cells and supports their recovery from stress. So in that sense, I would say that we have a unique approach. Also, there's this related stress responsive pathway that where there's quite a bit of activity now with, for example, AbbVie and Denali, who are developing compounds for integrated stress response pathway. It's related to the UPR, but again, it's more a single target pathway, and we're modulating the whole system. So I think we may have an upper hand there in that sense.
Thank you, Henry. And can you discuss in more detail the biomarkers that you have selected and why you have selected them?
Yeah, okay, so we explored actually a set of more than 90 biomarkers in the phase 1 trial with CDNF, and it included both PET imaging, but also alpha-synuclein, a set of biomarkers, and proteomics biomarkers, a set of 50 proteins in the spinal fluid, and also cytokines in the spinal fluid. And we did identify a number of biomarkers that changed with exposure to CDNF in a consorted fashion. And it's interestingly enough also these set of proteins reflected the mode of action of CDNF. So for us, this is a first indication that we have what we call a target modulation set of biomarkers. This will help us in further clinical development going forward into new studies. It will answer questions, for example, in relation to dose range, but also do we have an effect on the biology of the disease. So we are exploring, we are including the biomarkers that we have identified, and we're exploring also novel biomarkers, and we do need to do some validation work. So yes, indeed, we are implementing biomarkers going forward, and will help us answer a lot of questions, and it will make our clinical development much more efficient going forward.
Thank you very much, Magnus. And I think this is probably a question for you, Craig. How large is the potential market for X-CDNF and CDNF?
Well, the total neurodegenerative market is north of 60 billion projections by 2026. Parkinson's itself, there's 7 to 10 million patients worldwide that suffer from Parkinson's. And independent estimates of the Cdnf and ex-Cdnf opportunity suggest that the market that we could access would be in the order of about $8 billion for Cdnf or ex-Cdnf.
And I have my final question here. Based on the genetic and biomarker data that has begun to emerge, which we have seen more and more companies going down that route, particularly within neurology, which has traditionally been a tricky area to develop drugs in, and to focus on particular subgroups of patients based on genetics, phenotypes, and so on. Is that something that you are considering potentially looking into?
Yes, Kajsa. So I think it resembles the question that we received from Redeye. Indeed, we are looking into this, identifying a sensitive subgroup, for example, individuals with a specific Parkinson's disease genetic risk variant or gene variant would be very interesting to us. And here we have an indication from the phase 1 trial where we had this very interesting extended clinical response in an individual with this LRRK2 Parkinson's disease pathogenic gene variant. And it was not only an extended clinical response, but also the biology, the biomarkers moved in a consorted action, and we had a stabilization of the PET imaging signal. So truly very interesting, clinically significant response in this individual, which sort of puts the focus on this LRRK2 mutation patient with Parkinson's disease as a potential subgroup prone to respond to CDNF, and thereby something that we would pursue going forward with our clinical development of CDNF.
Well, thank you very much, Magnus, Henry, and Craig, for answering the questions today.
Thank you.
Thanks.
So I just want to remind the viewers that it's possible to watch the presentations afterwards. It will be on our YouTube channel. And thank you very much for joining today.